Hi, everyone. My name is Denghui Chen. I'm currently a research data analyst from Promet Lab at UCSD. And then today I'm going to talk about a new genotyping pipeline for Peromyscus californicus based on low coverage whole genome sequence. So when you guys see the title, the first question might pop out in your head is why Peromyscus californicus, aka so it's also called California mice. California mice, from the graph here, you can see actually phylogeny is a model between phylogeny is a really good species intermediate between two major rodent genetic models and humans, and it can serve us it can help us to understand the linkage between mouse and neuritis by serving as a really good outgroup. And then second, it is an emerging model for a number of psychological and behavior traits. For example, it is widely used for behavior studies, especially in autism. And also it is really good model for type 2 diabetes because they are monogamy, and it's also a really good model for partner fertility studies. Third of all, it has many well-established colony for other animals for experimental purposes, such as the PGSC. And so the only reason why it is not really widely used for genetic analysis is because there are no practical model methods for dense genotyping in this species. So from our lab, we have already developed a really successful genotyping pipeline based on the low coverage WGS for HS rats, SD rats, and then zebrafish. Here, we are trying to take over that pipeline and apply it to California mice. First of all, I want to thank all these people who supplied us all those initial 96 animals. And on the right side is like the genotyping pipeline flowchart for our genotyping pipeline. So let's jump right in. There are four main steps. The first one is the sequencing, and then the demultiplex. So for sequencing, we use for sequencing DNA isolation, we do 96 sample per batch. And then for the library preparation, we use the Riptide library preparation kit, which is based on random priming from both ends. And then it's also 96 animals per library. And then after that, we use the Illumina 9166000 to do the sequencing. For the sequencing process, we do 10 libraries per lane. So per lane, we do 960 animals. So it's like really high throughput. And then meanwhile, we also get like a 0.4 mean coverage for the WGS. And the software that we use for the multiplex is called F3 Bio, which is very specifically developed by the company who are developing the Riptide library preparation. The second step is the alignment. So alignment, we use the BWA-MEM, which is like a really general step. For the reference genome, we did have some issues about the reference genome, because the currently available reference genome from NCBI is only assembled at scaffold level. And then it's constructed with like so many different small pieces. It's not assembled to like a chromosome level. So it was not perfect based on our needs. And huge thanks to all these people from UC Davis who helped us to prepare a new reference genome for Peromyscus californicus. Although currently the new reference genome is still at the scaffold level, but they're still working on it to stitch them back to the chromosome level, and then it will be published to NCBI very soon. And then from here is like a basic comparison between the new reference that UC Davis is preparing and also another species from Peromyscus, which is really close to the one that we are studying right now. On the right side, you can see the current one, the current reference genome, the new reference genome that UC Davis is preparing. They're still at a scaffold level, but comparing to the to another species which has the reference genome for the chromosome level, we can see each scaffold actually covers one complete chromosome or multiple in some other cases actually. So which means the scaffold from the new reference that we have right now is actually very good, but they're still working on it to perfect it. And so based on the new reference genome that we have here, I'm showing to you guys two box plots. On the left side is the total of number of reads for each animal that we have, and then the right side is like the ratio of mapped reads. You can see the comparing to the old one, the new one has like a big improvement here. That's why we go for the new one. We totally abandon the old one, and then we hope the new one will bring us better results. And then the next step on the genotyping pipeline is the variant calling. So we follow the GATK best practice to do the variant calling. I'm not going to go into the details, but here's the result. We are able to call 28 millions of variants and then 23.9 million of SNPs, and inside there are 23.2 biallelic SNPs, which is pretty good based on the initial 96 animal that we have. And here's a PCA plot based on the 96 animals. From here, you can see the green dots. Actually, these 8 green dots, they don't cluster together with the with other 88 animals. It was when we look at it, it was because of those 8 green dots are from Glasper lab, and then they were caught in the wild, not bred in the. They were not bred in the from the colony. So we decided to drop those 8 animals because we are still at the process of developing and optimizing our pipeline. We want to reduce the noise as possible. And then after we remove the 8 animals from there, we still get 21.6 million of biallelic SNPs, and the PCA plot looks much better. And then, yeah, so the next step is the imputation step after the variant calling. For the imputation step, we use the software called Stitch, which is sequencing to imputation to constructing haplotypes. It's developed by Robbie. It's developed by Robbie Davis from Cambridge, and then you can visit that paper. This is like a software for imputation without a reference panel. So because we don't have a we also don't have a reference panel for the California mice. That's why we use this one. And so actually before the imputation step, we did some filtering because we want to keep like a set of high confidence biallelic SNPs before we do the imputation. So we also follow the GATK best practice to do the hard filtering for the variants. After, I'm not really gonna go into the detail about these plots, but this is showing the distribution of the filtering criteria that we did on the SNPs. And then after filtering, we still have 19 million biallelic SNPs, which is like a really good number for us to work with. Here are the density plots for 22 relatively longer contexts that we found from the reference genome. You can see they are really, they're sort of uniformly distributed among the chromosomes, which is good. And then a few small issues that you may see, I pointed out here in the small circles. Here, we see like a small gap here on the contact number 6. And then we also see another small gap on contact number 15. Actually, if we go back to the, these 2 small gaps, and also here on that plot, we see there's like a huge peak on the around location 12 0.5 megabase. And then we think the possible reason why we see those issues is because of the new reference new reference genome. They are not at the chromosome level yet. And then there are some cases where multiple chromosomes will stitch together to be one big scaffold. So that might cause the problem of the gaps here. And also there are some inverted chunks inside a contig that is from the new reference, and also there might be some repeated regions inside the contig actually. And then here are the rest of the contigs, the SNP density plot. And on the right side, you can see the number of biallelic SNPs for each long contigs here. Yeah, the number looks good. And then, so yeah, the number looks really good. We get like a lot of SNPs per each contig actually, based on their length. And so we also did some error estimation for the pipeline. So first of all, I want to say this project is not funded, so we didn't have enough money to do like a deep sequence to construct like a true set for this group of animals. What we did was we chose 16 out of the original 96, actually 88 because we got rid of it. Yeah, we chose 16 of them out of the original 88 to construct like a true set for our data. And then we try to match the distribution based on the colony and then the gender. So here, oh, actually this is 16, sorry. Yeah. So yeah, so the error estimation design is when we do the imputation, we do two runs. The first run is the original 88. And then the second run, we swap the original 16 that we chose for the control and then use the control to do the imputation again. So that we can compare the concordance rate between these two data sets. So as we saw, we have like so many different contigs. So we chose one with the length relatively long from those contigs. And then we chose contig 3, which has 154 megabase length. And then the number of biallelic SNPs is around 1 million. Concordance rate between the control and the original data set here is 81.4%. Yeah, this number doesn't sound great, but if we remember the reference genome issue that we talked about, if we remember the reference genome issues that we talked about, this is like a good number for us to start with. So from here, I think the possible cause for the low concordance rate is because of the problem of the new reference genome. And then we have like a really small sample size for the imputation software. Because the imputation software here that we're using is like a data-driven, is a highly data-driven one. And then the imputation, the other thing is the imputation software requires a lot of parameters. And then some of the parameters were based on the number of generation and then the funder information. And then we don't actually have like a very accurate and correct one for this data set. And the other thing is we don't have a reference panel for the imputation. It's a reference panel free imputation software. So with that being said, we think the next step that what we can do is we're gonna wait for the UC Davis group to refine the new reference genome so that we can have like a complete and better reference genome to work with in the future. And the other thing is we're gonna keep optimizing the pipeline to prune the parameters for GADK variant calling that we use, and also stitch imputation. Yeah. With that being said, here's the conclusion. So this is probably the very first cost-efficient and high-to genotyping pipeline for Peromyscus Californicus. And then it allows us to open the door to genome-wide association analysis for this species in the future. And yeah, that should be all. Before I close the slides, I want to acknowledge all these people from our lab who provide me a lot of support for this project. And also UC Davis, and then UC Davis people and UC Maryland people who helped us to provide the 96 initial animals, and then the people from UC Davis Genome Center who helped us to prepare the new reference genome. Yeah. Thank you for listening. Any questions?
Here's a quick one, I hope. The minor allele frequency for your 20 million plus variants, what does that look like?
It's actually pretty uniformly distributed along the, pretty uniformly distributed besides like the first peak at the very beginning, I think. I can show you a small plot, but this one is not like the scaffold that we discussed about. This is from another scaffold, but the top here is the malady frequency distribution.
If you're interested. Very even. And that population is from within, what's the capture zone? 100 miles less?
I'm not sure about the how they collect the samples, actually. I'm sorry.
Okay.
I cannot answer this exact question, Rob, but it is a lab population which was established approximately 80 generations ago, quite long ago, from 60 founders. And I don't know how the 60 founders were caught. And then this original population is an origin for trainer lab and Fisher lab colonies.
So many of you already know from yesterday that the new rat reference genome is MRAT-BN7. This new reference genome was released November of last year, and it was sequenced using a single brown rat from Medical College, Wisconsin. And sequencing and assembly were done by the Sanger group, coordinated by Chen Hao. And the brown Norway is the same strain as all previous rat references. And this new version is now available from all major rat genome databases, including the RGD and CPI Ensemble, UCSC. UCSC calls it RN7. And the official name is MRAT-BN7. And occasionally you might also see people talking about MRAT-NOR1, and they're all the same. Even before the release of BN7, a group of loosely organized as the International Rat Omics Consortium started to talking about rat genomes. By the way, kudos to Boris Topakov to come up with such a cool name, IROC. It is really an honor today for me to highlight some of the great work done by the group in the past few months that compares the new reference with the old reference, and also pointing out a few potential remaining issues in the new reference. So Wendy from yesterday's talk pointed out that the new reference has much improved contiguity compared to the old reference. In addition to that, the new reference also fixed many structural issues. On the right side here is a dot plot of chromosome 6.
This is. There's also an open question as to what local means for regulation. Usually this is defined as a region spanning 100 to 500 kilobases flanking the gene on both sides, but there's no hard agreement as to why biologically this happens. We had some basic questions we wanted to answer with this project. Do the various kinds of local regulatory elements interact with each other to any quantifiable degree? And can we meaningfully define what local really means in the context of regulatory interaction? And furthermore, what can this teach us about stem cell state in genetically diverse model systems? And we've got some results that I find quite exciting, a small portion of which I'll demonstrate in the regulation of a gene called Platter2. Platter 2 has recently been implicated in the regulation of mouse embryonic stem cell state, and it produces a long non-coding RNA that's transcribed in embryonic stem cells, and genetic variation in and around this gene has been observed to have distal regulatory effects on a number of embryonic ectoderm-associated genes in stem cell culture via eQTL and mediation analyses. So to dig into how all these questions about local regulatory area that we have and genetic epigenetic interactions and our example gene, we decided to employ a panel of diversity outbred mouse embryonic stem cells created by predictive biology from Jack's mice. So these are similar to the collaborative cross mice that were mentioned earlier today, but these are outbred heterozygous populations of unique individuals rather than a set of homozygous strains coming from these 8 founders. These mouse embryonic stem cells derived from the DO population were grown in a one inhibitor medium that exposes a number of cell phenotypes not normally seen in stem cell culture. And we have bulk RNA-seq, TAC-seq, and haplotype for 176 of these samples. And these data have already been used for eQTL analysis to identify regulatory SNPs that could alter cell colony morphology. So that's with these people here. So that would let us look at whether the genetic variants in these cultures interact with open chromatin to affect gene expression. And to survey this on a broad scale, we used regression modeling, which is a statistical technique that permits us to determine the most likely relationship between each SNP, attack peak, and gene. And we selected only those trios of elements that were predicted to be interacting and had a Bonferroni adjusted p-value of less than 1 × 10⁻⁷. So all the graphs you see from here on out are only looking at results that hit that quite conservative threshold. Now, onto Platter 2. Attack-seq peaks are easy for us to map accurately, so we looked at which peaks are involved in interactions with this gene and how often. We see a pretty interesting distribution here with interactions appearing more than 500 kilobases away from the gene. And there's also a dead zone here, followed by a little distal cluster of interacting attack peaks that pop up 1.5 megabases away from the gene. And this holds true for all of the genes in the TAD. And we found distributions like this all over the place. There were dead zones followed by distant interactions that affected gene expression. So what's going on there? Well, this is where the physical structure of the genome comes into play. In birds and mammals, DNA is arranged into loops called topologically associating domains, which are pinned together by the CTCF complex. Inside them, transient subloops bring regulatory features into contact with genes. And CTCF insulates TADs against outside interactions. Many are stable across cell types and conserved across species, and have an average length of about 1.09 megabases in mice, which puts it within the range to either contain or constrain the length of a gene's local regulatory region. So we pulled a publicly available data set from the Bing Ren lab, which gives the approximate coordinates of TAD formation in C57 black 6 mouse embryonic stem cells, and we overlaid them on our data. And so these long TAD loops transform our distribution of regulatory interactions from this into this. And suddenly you're not looking at a mysterious dead zone and distal clump of interactions. It's the continuation of the regulatory regions found on the other side of the TAD boundary. And we see this again across the genome when genes are influenced by regulatory interactions in regions placed closer to them in 3D space by CTCF-bounded TAD loops. And in fact, when we look at the TAD boundary locations across the genome, we see a strong preference for interactions that occur inside the same TAD as the gene being regulated versus outside of the TAD. Now, back to platter 2. We're able to get more information about its regulation from our interaction analysis, which further illustrates the role of TAD formation in gene regulation. When we break down the SNPs and attack peaks that interact to affect platter 2's expression levels, we find that 95% of these interacting elements are not co-localized. So these aren't just physically overlapping to cause interaction. And this also means that if you ran a differential expression analysis or EQTL analysis on this gene, you probably wouldn't be able to draw the connection between these regulatory factors. But that just tells us that there's interactive regulatory activity here, and it's with consistency across our samples. What other information can we extract from this analysis? Well, one aspect of this modeling approach I haven't touched on yet is the beta coefficients, which are often referred to as the effects of each model term. And these are constants that are calculated for each trio of gene, SNP, and attack peak to create the best fit for the data that you give it. And these effects can't always give you precise information about fine distinctions between models, but they can give you more general trends, like functional categories of regulation based on the pattern of positive or negative signs of these effects. So positive effects are more associated with more transcription of a gene, and negative effects with less expression. So when we look at the effects within these models by direction, we can split the results into functional groups. If all the effects point in the same direction, the SNP and attack peak are working in synergy with each other. If the main effects point in the same direction, but the interaction effect points the other way, that indicates functional redundancy in their mechanism of action, or that they're interfering with each other. The remainder are hard to interpret, but the relative magnitude of these effects in these models indicate that there's probably multiple functional groups within each of these categories. So when we look at platter 2, we see redundancy and interference dominate the results, especially when compared to results for all genes we have data for. This tells us that if we wanted to alter platter 2 transcription rates to test how it contributes to stem cell identity, it wouldn't be as simple as selecting the right genetic variant for the job, because there are potential compensatory mechanisms that have to be accounted for, and but this is a good example of the kind of regulatory complexity that maintains homeostasis in genetically diverse populations, where maintaining expression levels at specific amounts can be crucial for development and normal function. Now, there's one more thing about the platter 2 TAD that I want to point out, and it's the interacting regulatory features in this area here, where the TAD boundary lies. There are 29 interacting attack-seek peaks in that region. And when we look at what sequences are actually contained within these interacting attack-seek peaks, we find that one of the motifs that's overrepresented is the SMAD3 binding site, which is a component of the CTCF binding complex and is a regulator of TAD formation. So this potentially suggests that platter 2's activity may depend on the stability of its TAD or in adjustments to its boundaries. And other researchers have previously demonstrated that the modulation of TAD boundary location is a vital regulator of mouse forelimb development and human epithelial differentiation, as well as immune activation and transdifferentiation. So we're not only seeing that tab loops define the local regulatory area of a gene, they're also an active regulatory mechanism, and we may see evidence of this regulation around platter-2. So in summary, we find that we were able to predict the locations of interactions between genetic and epigenetic regulatory factors across the genome in diversity outbred mouse embryonic stem cells. We found that the location of these interactions is bounded by topologically associating domains, and that intra-TAD interactions are a good place to start looking for interesting biology, such as with platter-2, which is under active regulation by multiple redundant regulatory factors that we speculate could provide compensatory backups to maintain gene expression at appropriate levels. And for those who are interested in applying these kinds of principles to your own data, but you're missing one or more components of the analytical method used here, the distribution of where we found interactions suggests that you can capture 95% of all the interactions we've found within a 3.9 megabase window centered at the gene start coordinates with 1.84 megabases upstream and 2.04 megabases downstream. And that's it. Thank you all. And I'll be happy to take any questions.
That's great, Lauren. Thanks very much for that talk. We had to work a little bit into our break. Sure. Because the next talk is going to be at 12:30. So we've got 11 minutes. You can either use that for continuing the discussion or taking a break and getting some refreshments. But our next speaker is going to be Mikhail Maksimov at 12:30.
Okay, wonderful, perfect. Okay, hi everyone again. So first, a big thank you to the organizers, of course, for accepting my talk and giving me this opportunity to present at this awesome symposium. My name is Mikhail, and I'm a postdoc in Melissa Jimrich's lab at UCSD. And my talk today is about short tandem repeats in the BXD mouse family, how we're doing QTL mapping with those. So work in Melissa's lab is focused on understanding how complex genetic variation contributes to human disease and what kind of impact it has on gene regulation and complex traits. My project's a little bit different because this is the first time we're looking at tandem repeats within a model organism like mouse. So it's a cool opportunity to do genome-wide characterization of STRs in a highly relevant model organism population and to demonstrate how we can leverage this type of information to learn about new biology. Okay. So very briefly, STRs are head-to-tail repeating DNA elements with the motif length between 1 to 6 base pairs, so like short elements. And we're using a reference-based method to genotype these repeats. And because of that, we first need to have a list of STR loci that exists in the mouse reference. So then we're taking short-read Illumina data for BXD strains, which that we get from the BXD sequencing consortium, and using that to infer maximum likelihood repeat counts at each strain and at each locus. So for panel construction, basically, we're running tandem repeat finder to generate the list of predicted STRs. And then we just apply some filtering to get rid of like imperfect repeats and very short repeats and bundles that likely our tool is not going to handle well. You know, like the total number of repeats in the panel is going to vary somewhat based on how aggressive we do this filtering. But roughly we find about 1.2 million loci in the autosomes in MM10. Not showing this here, but we actually saw a dip in call rate going even from trinucleotide repeats to dinucleotide repeats. So we actually chose to exclude the homopolymers from this analysis at this point. So I'm just focusing on like tumors and everything that's a little bit longer. But that still leaves us with a good number of STRs to analyze. And in fact, after filtering out monoallelic loci, because remember that just because these exist in the reference, it doesn't mean that there's any variation among the actual BXD strains. So then we're left with about 76,000 segregating STRs. And let's see, a couple ways to check that our STR calls actually make sense. So first, we're able to replicate the patchwork pattern of inheritance of SNPs using STR genotypes. I'm just showing one chromosome as an example here, and just color coding by the haploblock, the founder inheritance in STRs, and then you can see how basically they match really, really well. So that's the first thing that gives us confidence. And also, we've run a small validation study using capillary electrophoresis. I'm just showing a summary on the right with about 100 loci and 16 strains. And we're able to validate close to 90% of the calls. We did have one problematic locus where we were off by one repeat unit in several strains, but this was one of the more complex loci, where basically they're like two STRs that are pretty close to each other, almost like overlapping. And so that's obviously like fairly challenging, but everything else looks pretty good. And one of the motivating factors for studying STRs, of course, is like the interest in the number of diseases that are known to be caused by repeat expansion, and also microsatellite instability. And there's been a large number of disease-focused studies looking into like molecular mechanisms of MSI, and also the involvement of mismatch repair proteins in regulating STR stability. So like in the case of cancer, a lot of times this is done in the context of somatic mutations, but inbred mice can actually help us look at genetic effects, because confounding factors such as like the age of the father, for example, or other environmental factors are more minimized. So to this end, we knew we wanted to identify and look at new STR variants that arose in the strains. And we basically just took a pretty straightforward approach to do this. We just simply compare the RI strain genotypes to founder genotypes every locus, and note any RI strains with a genotype that's different from both of the founders. So then we take those and we overlay it on top of the B and D haploblocks, founder haploblocks that we get from SNPs, and assign the founder of origin from which this new variant arose. And what that allows us to do is to calculate the magnitude and the direction of change in the repeat length relative to the expected founder. So as you can see in the bottom right, we found about 18,000 loci that have a new variant in at least one strain. And most of these are tetranucleotide repeats. And this actually just mirrors the motif-like distribution among all the segregating STRs. So in terms of like properties of these new variants, as expected, we have a pretty nice correlation in the abundance of new STR mutations with the number of inbreeding generations that the strains have undergone or been subjected to, I guess. Top panel here shows the abundance of new variants per strain for each epoch. And most of the repeats in the bottom left, the contractions or expansions, it's just a plus or minus one repeat unit with some larger ones as well that can happen. And the majority just appears in one strain. So like as expected, most of these new variants are singletons, but there are a number of variants that are, we think, are shared across a number of strains within like a particular BXD epoch, as evidenced in one case by the separation of strains into like epoch-based clusters in a PCA type analysis using like the presence of the new variant as an indicator variable. So we get some separation into epoch-based clusters there. And we can actually see that some of the variants are in multiple strains. It's the exact same variant. So that's sort of like the properties of these new variants that we found. But then to actually get to investigate mutational properties of STRs, we need to come up with some metrics that we could run QTL mapping on, and we decided to look at the different quantitative phenotypes. So being mutation rate, mutation size, and propensity to expand. The rate is just a proportion of genotype loci that are new variants in the strain. And the mutation size is the average size of increase or decrease in the STR length in repeat units. And then the expansion propensity is a proportion of new variants which are expanded in the strain as opposed to being contracted. So for each of these phenotypes, we filtered out epoch variants before QTL mapping or before calculation of the phenotype. And this is just the reason being that these first more likely to be false positive given like an incorrect founder genotype assignment. If we start to see an epoch variant where like the majority of strains are supposed to be new, then that's a little bit weird. And also second, some of these that are true epoch variants could have been segregating in the number of strains that are different from the individual where the variant arose. And so that probably wouldn't help us very much. So after we had these phenotypes, we could actually get into running the QTL mapping. And so here I'm showing some results for this. Basically, we ran QTL mapping against, for each of the phenotypes, each of the mutator phenotypes against a list of 7,000 LD independent markers, basically directly from Professor Williams's lab, downloaded from GeneNetwork. And we used linear mixed model with a number of inbreeding generations. We used as a covariate because we actually see even like within the abundance phenotype a substructure by epoch. And strain kinship matrix, just the standard kind of approach to control for population structure using the RQTL2 package. So we didn't find any strong signals for the mutation rate or mutation size phenotypes, but we did find a pretty robust looking QTL signal on chromosome 13 for the propensities of STRs to expand. And we did go back and check that this result is robust to selection of epoch variant filter. So let's just try to filter out, just to try to set a slightly different threshold and see if this is replicable, and it is. And one of the things that we thought could be interesting is if this expansion propensity signal was driven by a specific type of STR, which could be actually like a biological reason for that. So what we did is that we repeated this QTL analysis, but for different repeat classes separately. And what we saw is that the strongest signal actually comes from the tetranucleotide STRs. But in our case, it might just be due to sample size, because the way that this has worked out in our data set, the tetranucleotides are the most abundant repeat class. So actually for all of the kind of like finalized, optimized QTL mapping traces, I'm just showing with those two filters applied, just using tetranucleotides and having the specific epoch variant filter. Okay, so next to try to understand this potential candidate QTL a little bit better for mutator phenotype, we know that it's centered around 91 megabases, but the confidence interval is pretty wide, around 10 megabase window. So to characterize it a little bit further, we wanted to look at the genes that were in close vicinity to the peak center. And there's about 50 confirmed genes in the region, not counting like the various predicted ones that there's not much information about. We didn't initially find any variants that were predicted to severely impact protein function. So we thought that perhaps this effect is mediated through gene expression. And to investigate this, we ran now eQTL expression, gene expression QTL mapping using the data available from GeneNetwork again. So here I'm just showing like a heat map, basically with value being the maximum eQTL log odds score in this small, or in this like 10 megabase region for every gene, a data set pair that we analyzed. And the genes are just shown on the x-axis and sorted from left to right by the number of data sets where the peak eQTL exceeded the permutation-based threshold. So basically, like the genes that are to the left are the ones that in more GeneNetwork data sets have an eQTL signal in that same place where we think we have our QTL signal on chromosome 13. And we selected, to be able to make the data a little bit more manageable, we just selected one representative data set per study that's deposited in GeneNetwork and try to get as many tissues as possible to get a more comprehensive view, which the tissues I'm showing on the left. So from this heat map, I think we see some candidates appearing that have co-localized signal, eQTL and QTL for the mutator phenotype. And among these, actually pretty interesting, they have 3 genes that are involved in DNA repair. So XRCC4 facilitates nucleotide excision repair, and SSBP2 is single strand binding protein involved in the damage response. And MSH3 has been widely implicated in STR stability in both mice and humans. So there's actually quite a bit of research about the role of MSH3 in repeat expansion. So this is our favorite candidate at the moment. So one things we did next is we just plotted our phenotype basically again. We're back to the phenotype and just plotted it by and color coded by founder haplogroup at the peak EQTL. At the peak of the EQTL. And what we see is, as the top left panel, is that the strains carrying the B haplotype in this region, they tend to have a higher expansion propensity than the strains carrying the D haplotype. And then similarly, for each of the DNA repair genes, it's the bottom right panel, we also see increased expression associated with having a B founder haplotype, which I think just from the other studies that have looked at MSH3 in the context of repeated expansion, this trend makes sense. In the top right, I'm just showing where these genes are located relative to the QTL peak itself. And then in the bottom left, the EQTL traces, the top 3 candidates, and the many different traces with like different degrees of opacity are the different gene network data sets. And then the black is just the actual mutated phenotype QTL, just showing that these are co-localized pretty nicely. So in summary, we characterize the STR repeats that segregate within the BXD family. So that's a win number 1, and identified the number of new variants, and then calculated 3 quantitative mutator phenotype metrics. And I also hope I presented some compelling evidence for the presence of this STR mutated QTL among the BXDs, and for the propensity of STRs to expand rather than contract. We have a few candidate genes at this point, one favored gene that make a lot of sense biologically, for which the QTL signal and the QTL signal localize. And from here, we still have some work to do, I think, in identifying the causal variant, which would be really cool. To that end, we're currently like hunting down various leads with structural variants in the region. So that's an ongoing process. And so with that, I just want to really thank everyone for their attention and acknowledge all the colleagues that support me in this research. Of course, my advisor, Melissa, Nicole, and Nima in the lab. And Nicole ran the whole PCR validation experiment study that I presented. And then Nima has been a great resource for the genotyping tools. He's like the expert in the lab who came up with the method with Melissa. Of course, I want to thank Professor Palmer for all the advice on this project, and then Milad in his lab. I frequently ask him various questions on how to do things. And of course, Professor Williams, David at UTHSC for bringing us into the BXD project and the expertise and guidance in this. And I want to acknowledge that the funding for me for this project is from the NIH. And of course, I'm happy to take any questions if there are any.
Hey Kyung Im, I'm not sure if I pronounced your name properly, I'm sorry, but you're from the University of Chicago.
Yes, pretty close. Thank you. People call me Haki, which in the US, people pronounce it like the sport, the hockey. Anyways, so that's some tips on how to pronounce my name. So can I share my screen?
Yes, please.
Let's see. Okay. So do you see my presentation?
That's perfect. Yeah, great. Thanks.
Great. Thank you for the invitation to talk today. Let me start with the acknowledgement slide because I tend to forget it and it's really bad. So, okay. So the work I'm going to talk about was done by the people listed here, Natasha, Sabrina, Tyson, and with the help of other members of the lab listed here. A. Palmer was a critical collaborator who made this possible. he gave us access to the data, the rich data set generated by the Center for GWAS in outbred rats. I tried to list everybody, but if you are among the more down here, forgive me and let me know, and we'll list you when we write the corresponding paper. Okay, so this is something we all know. Genome-wide association studies in humans have discovered hundreds of thousands of loci, and we keep discovering more every month or year. But most of those variants that we discover are in non-coding regions, so the underlying or the mechanism behind these loci are still not well understood. So the general consensus, though, is that the transcriptome has a mediating role in that link between the genetic variation and the phenotype. So in a GWAS, we associate DNA variation with phenotype. And what we propose based on this idea, this was back in 2015 when the paper was published, was the PredictScan framework, where we proposed to use mRNA levels, so inferred mRNA levels based on DNA variation, associate those inferred mRNA levels with the complex trait to try to understand, find the causal genes, or at least prioritize the list of, reduce the list of causal link candidates, causal gene candidates. So the way we do that is, please interrupt me with questions if there's anything that is unclear. Okay, so how do we go from DNA variation to mRNA? Usually, mRNA assays are much more expensive than genotyping. So what we proposed to do was to use these reference genotype and transcriptome data sets such as GTEx in humans, take the genotype data, the transcriptome data, and fit this linear penalized regression models where we take one gene at a time and find these weights that allows us to predict or impute the level of expression so these different columns in the transcriptome data set using the genotype data. So we train these weights using different approaches, and we save those weights in a database we call Predictive. So once we have that, we can go to a GWAS data set where we have genotype data and we have trait data. So what we do is using the weights that we kind of trained previously in the reference transcriptome data sets, and from the genotype, we generate this predicted or imputed transcriptome. And then we correlate the trait with each one of these columns, which are predicted transcript levels, and that yields what we call PredictScan results. That's this table of where you have genes in the rows and then effect size, standard error, and p-values. And small p-values are significant associations. So if you have questions, please ask, because I can't really read the chats right now. Okay, so how well can we predict these genes? So in humans, I've selected here 4 representative in the sense that these are highly well predicted genes. So ARAP2, NADI2, PEG6, and ARAP1. And you can see on the x-axis, I'm showing predicted expression. On the y-axis, I'm showing the observed expression. And you can see that the R squares, this is correlation squares between predicted and observed, are pretty high. So for these genes that were selected for being well predicted, you can see that predicting from genotype transcript levels is possible. And more generally,
actually, if we go back to the last slide, can you clarify for people who might not be used to thinking about it? The upper left one, we can see the genotype classes that you might expect, I think, right? But some of the others, there's not defined genotype classes here. Is that because there's many SNPs? Is that right?
Exactly. Actually,
to help people think about it.
Yes, thank you. So we use, and I'll go into more details later, but we allow the statistical model to choose however number of SNPs are optimal to do the prediction of the transcript levels. And you see for this gene, ARAP2, it seems that it's just one SNP, right? And in effect, there is a causal SNP for this gene that is known. The thing is, our prediction model here has 80 SNPs. And in fact, what these 80 SNPs are doing is they are predicting this other, imputing this causal SNP that we didn't have in our prediction set. Anyways, that's, I hope that clarified rather than obscured what I'm trying to say. And please interrupt me anytime. So here I'm showing you about 13,000 genes ordered by their heritability estimates. So you can see the black curve underneath here is the estimated heritability. The gray bars are confidence intervals. And in red dots, I'm showing you the R squares. So the genes that I was showing you before are somewhere in this upper right left, whereas there are other genes that are really, that transcript levels are not really predictable, heritable, so therefore not really predictable, and that's what we are seeing in the prediction performance. So this is all consistent. So summarizing quickly, what are the advantages of this PredictScan or TWAS framework? We have much reduced testing burden. Instead of testing millions of SNPs, we are testing about 20,000 genes. The function of genes are much better annotated than our SNPs. We don't have as much the issue of reverse causality because we are looking at the genetic component of gene expression rather than the actual gene expression levels. Another advantage is that it provides direction of effects. That means that whether up or down regulation of a gene increases the risk of a disease. The candidate causal genes should be good targets for drug development, and also transfer of knowledge across ancestries and species is easier using this gene level approach. Okay, so that was a review of what TWAS or PredictScan does, and now what we wanted to do is translate all these pipelines into the rat data. So we start by generating prediction weights for the transcript levels in rats. We used 80 rats, from 5 different brain regions. So this is the genotype matrix where rows are rats, columns are different SNPs, and the transcriptome data for the 5 different brain regions. So the prediction is, as I said before, is one gene. We predict one gene at a time with this linear model. So we did this and saved it into a rat predictivity database. So here is some of the results for the 5 different regions of the brain that we used. We were able to generate prediction models for about 8,500 genes. So the number of genes that we're able to predict is a sense of how well we can do, because with larger sample sizes, we tend to see more genes that are predicted. But in comparison to what we saw in humans, so we applied this pipeline to the GTEx data set, to the 49 different tissues available in the GTEx data set. And in that case, we were able to find, have convergence for 8,500 genes when we had a sample size of about 420 individuals. So in this data set, we only have 80 rats, so which indicates that the environmental component is smaller, or that the genetic component is larger in these heterogeneous stock rats. So that's a good thing for us. Here is just a list of the best performing genes. So here the R squares was calculated in a cross-validated manner, so this is not overfitted. We can see these are pretty high R squares numbers, and p-values are all significant. Okay, so how many SNPs were used in the prediction of the transcript levels? it's a median of 10 or a few more. So these are SNPs. We should not think of them as causal SNPs, but they do give us some sense of polygenicity, if you will. Okay, so similarly to what we did in with humans, I'm showing you here again. So we sorted genes by their heritability estimates. So we tried with both GCTA and BSLMM. BSLMM works slightly better, so that's what I'm showing you here. So heritability, so because they were sorted by their values, you can see this continuous line. And the red dots again are the R squares, so the correlation squared between observed and predicted expression. So here it's, the genes whose transcript levels are not very heritable, we don't do well. And on the extreme right, we see that those that have highly heritable transcript levels, we tend to predict better. So here is a comparison with humans. This was done with 922 individuals. So you can see that the red dots track the heritability estimates much better than we do here in this data set, but it's more than 10-fold more samples in this case. So I think we're doing pretty well. Just a summary of how well we predict. For the different regions of the brain, we have a median of 3.1% to 3.8% correlation squared, so R squares between predicted and observed, with a maximum between 81 and 83%. We compared the predictability in rats with predictability in humans, and it's not very clear. The plot is not very nice, but we do see, small 7% correlation, but significant. So p-value, that's pretty small. So this is telling us that genes whose transcript levels we were able to predict better in humans, we will also tend to predict better in rats and vice versa. We also saw that the predictability was similar across different brain regions, which is not surprising. And another thing we did, this was done today by Sabrina, was calculating the correlation between the genetic component of genes for these different regions. So we correlated the predicted or genetic component of gene expression for all these different tissues, and the correlation is above 57% across the board. So that's telling us that the genetic components of transcripts are correlated across tissues.
Kyung, I'm sorry, we've run out of time. Would you mind to be brief and trying to wrap it up, please? That'd be great.
Yeah. Okay. Yes. Yeah. Okay. So since we have the prediction models, we apply them to 440 different traits that we were given. These are some Manhattan-like plots that we were able to generate. And let me go directly to the summary. So, take-home message. So we are translating the human PredictScan TWAS framework to rats. What we found was that the genetic architecture of transcript levels in rats is similar to humans. Transcript levels in rats are heritable, so genetically predictable. Heritability and predictability in humans and rats are correlated. The genetic component of transcript levels are correlated across tissues. And we found some significant gene trait pairs that we will continue to analyze further. Okay, that's it. Thank you.
Lovely. Thank you very much indeed. Because we've run out of time again, could we have questions in the chat, please? And hey, just have a look at the chat and see if there's anything for you in there.
We'll do.
In which case, great. Could I please move on to our next speaker, Vivek Kumar from the Jackson Laboratory. Vivek.
Thank you, Reinmar. Are my slides visible?
Yes. Yeah, that's
great.
Perfect. Great.
Great, thanks. So thank you for organizing this, and thank you for the opportunity to talk here. Let me just start with some credits. The bulk of this work that I'm going to show you is done by a set of collaborations we have with Indian Institute of Technology. And the student here is Dhruv Vlad, and the mentor is Balaram Ravindran. And I would also say that most of this work is really a work in progress. So the details may change. Hopefully the take-home message will stay consistent. Needless to say, the neuroscience, behavioral neuroscience specifically, is being transformed by technology. This is over the last several years, advances in statistical learning have made their way through to biological applications. We, along with many other labs, are applying these methods. Our core principle comes from the BRAIN Initiative. We're interested in automated quantification of behavior at high temporal spatial resolution, reliably, objectively, over long periods of time under a broad set of conditions, and in combination with concurrent measurement and manipulation of neuronal activity. So this is an emerging field of computational ethology. Broadly, the goals that we've set forth are to ethologically monitor outputs of neural circuits, which is behavior. We want to do this over long periods of time with high resolution with multiple animals. And we want to manipulate the environment, the nervous system, through genetics, pharmacology. Before, really, the first part is challenging, which is just how do you annotate behavior at high resolution? And the field, we've taken several approaches. The main three are described here. So the classic approach is heuristic method. Animal goes from the center to the periphery, or periphery to center, and you call that a crossing. There's not much interpretation involved. We just look at the animal tracks. The newer approaches are using supervised methods of classification and unsupervised classification methods. And the underlying assumption of our approach is described here, which is to obviously use the mouse as a model for human conditions and diseases and interventional therapeutics. We share genetics, we share our neural circuits, and that's in this purple box. These are conserved elements between us. What's not conserved is our environment. So the brain is an organ that's evolved to really take in sensory input from the environment to optimize behavior in order to have the best ability to reproduce. So the environment that a human lives in and a mouse lives in is very different. So it's understandable that the behavior outputs from these two organisms are also going to be different. And what we have to do with this new technology is try to correlate across these two behavioral spaces. And this is the idea of phenologues coming from Ed Marcote's lab. He applied it towards kind of biochemical and developmental phenotypes in yeast and other organisms. We can apply it towards behavior. And I would argue that this is working its way towards this idea of clinical ethology. So just to give you an idea, the traditional assay, let's say, we have open field assay, traditional metrics are like total distance traveled, percent and corner. From this new methodology, we get a whole host of new metrics, such as we can get like very accurately measured gait, which you saw Gautam present yesterday from my lab. We can, quantitate the number of turns, the amount of grooming. If we have multiple animals, we can look at chase behavior, approach behavior, contact behavior. And many of these metrics are very subjective, i.e., we will not agree within labs and certainly between labs what a chase behavior may be. And when you take this out to kind of a further unsupervised behavior space, this is data coming from motion sequencing from Bob Dada's lab at Harvard. And there are just, a ton of these behavioral motifs that the algorithm produces, and we have to kind of work our ways we have to determine what's actually relevant. So we've been tackling this in a series of papers where we've looked at tracking, action detection, gait and posture, nociception, frailty, sleep states. And we've used this approach of either building classifiers or building rules, or kind of combining classifiers with heuristic approaches. And we immediately run into a problem. So I'm just using this as a toy example, but it's a real problem we have, which is here we're looking at a very simple behavior, left turn. We can all agree that we think we know when a mouse makes left turn. But the actual fact is, and here are two labelers, so you'll see for the most part, there's overlap. When this light goes off, that means the classifier from A and B thinks the left turn is being made. And what we find is that this is the actual path of the animal. Classifier A is really calling this as turn, and classifier B is calling this as turn. And this is a triple example, but this really, when you start looking at social interactions, gets really complex. And we start to disagree tremendously on what the behavior is. And so we started to build, we started to collect behavior classifiers. So the idea here is that a user will submit a classifier that's really expert knowledge that their lab has and that they have on what that behavior is. And we can then kind of share that classifier between labs. And now my lab doesn't have to really understand fully or learn how to annotate your behavior interest. We can just use your classifier. But how do you start to prioritize classifiers? If 3 people submit a left turn classifier, which one do you download and which one do you start to use? So we've built this app here, and underlying this app is any classifier that gets deposited. We have a data set of 2,500 videos across 2,200 hours. This is single mouse behavior with 62 mouse strains. And so immediately what we can do is calculate heritability from that, and we can carry out GWAS. And what we propose is that you use the classifier that's more heritable. Okay. And just to give you some taste for what we find, these are gait measurements, and I'm just showing you here heritability. Here on the left is the mean trait heritability, on the right is the variance heritability of that trait. And we get really good heritabilities. Where we get poor heritabilities up here are things like temporal symmetry, like or angular velocity. So when the nature has selected mice to walk in a symmetric way, which is not surprising. It's not. It doesn't vary in the strain population we're looking at. So we've been using this approach. We've now applied it towards unsupervised behavior space. So there are 3 kind of major methods of unsupervised behavior mapping. We use this derivative of this method called B-side from the Yitri lab at Carnegie Mellon. The process is fairly complicated, but we start with pose. We go through a series of feature extraction, that's temporal feature extraction, and then we use clustering methods to kind of cluster this. We end up with, and again, the data that a lot of labs would use is 1 strain or 2 strains. We here have data from 62 different inbred strains, 2,000 animals. So we really are getting a complete picture of what the mouse does in the open field. And so what we end up with is 61 behavioral motifs. 42 of these behavioral motifs account for 90% of the time that the mouse spends in the open field. These motifs are quite small, 100 to 200 milliseconds. So what we did next, and just to give you an idea, these are motifs like, for instance, here's a right turn. So the animal starts in one position and then makes a right turn, or here's a rearing motif, here's a kind of just walk in a straight line motif. So these are small millisecond long motifs. And not surprisingly, we can partition or cluster mouse strains purely based on their behavior. This told us obviously that these behaviors are heritable. We calculated, and this is SNP heritability from coming out of GEMMA, and we immediately, so some of the motifs are highly heritable. We're reaching 0.7, 0.8 in heritability. But you notice a lot of the motifs, about half of the motifs are have heritability of less than 0.2. And then the question becomes, well, the mice are spending a lot of time doing this, but what does it really mean? And I think this is where the project is right now. Now we have to, unsupervised is easy on the front end, hard is on the back end. Now we've got to figure out, well, what's going on with these. We have to actually look at the videos. And I think in the end, what we need to do is improve our algorithm and trying to eliminate some of these kind of low heritability motifs. I think we're just not getting enough information about what the mouse is doing, and that's leading to kind of noisy behavior annotation here. So that's kind of where we are with this. The idea, again, is to carry out unsupervised analysis, but then layer, and the foundation, though, of this unsupervised method is using heritability with this kind of large data set we have. And ultimately, what we need to do is start to connect these motifs to, as I mentioned in that phenologue idea, to disease states, to altered circuits. And that's where, that's where a lot of the work lies. And by the way, we need, I think, in the field, better ontology terms. The field is really, the ontology terms for behavior are old. And we really need to kind of revisit that whole ontological term database that exists. Yeah, so heritability as a filter for relevant behaviors, that's our major conclusion. Overall process of unsupervised learning works, but I would say it needs improvement. And then, 50% of the motifs are less than 20% heritable, and we can talk about how meaningful that is. Just to thank everyone. So again, Dhruv Lad did most of the unsupervised work here. Just to plug here, we're looking for postdocs, grad students, and even RAs. So if you're interested, please reach out. Thank you.
That's great, Vivek. Super, very fascinating talk. We've got some questions in the chat, and I've got a question for you as well. When you study these motifs, and you think about the sort of the biological relevance of these motifs, and what role do they play? I mean, were they either particularly selected for during the breeding process, or are they relevant in the natural behavior? What is it that you're trying to get at the end of the day by focusing on those motifs?
Yeah, so that's a great question. So the idea would be that these motifs, once we kind of get once we can define these motifs and we have kind of the repertoire of mouse behavior, then we can go into a disease model. We can go into a pharmacological perturbation model or optogenetic or human, some model. And then we can say, when you turn off this circuit, or when you perturb this genetic circuit or neurobiological circuit, now this motif disappears. So then that motif becomes a phenologue of something that you can, that's hopefully relevant to disease in humans. Does that make sense, Ryan Mar? That's the idea here. And of course, the power of unsupervised approach is that if, once you work out what these motifs mean, you can do this at scale, because this is very straightforward to do. Anyone should be able to do it. And if you collect your data to the same standards as we do, you can actually just, we can start to trade video, our motifs, our classifiers, all these things.
That's really fascinating. Thanks so much, Vivek, for that. There's a couple of more questions. Yeah, Rob, you've got one, quick one, yeah?
I think this is really important to make sure everybody gets this. It's really, I used a fancy word just to intimidate molecular biologists again. reify. So it's really important not to reify heritability too much. We know that selection is working to suppress heritability. It would like to eliminate heritability. That's why all of us, if we tried to measure bilateral symmetry, do we each have two arms? Yeah, pretty much. No variance. So you can't measure it because, and so paradoxically, some of the measures that have the lowest heritability are precisely the most interesting. So don't let the low heritability scare you, Vivek.
No.
That's what PhDs are for. And I just want to make sure nobody should come away from your talk thinking because it's low heritability, it's inaccessible genetically. It's not. You just need to control the environment or resample. Or else fools like me and Gary would not be doing studies of longevity in mice. That's a loser's game because the heritability we know is about 0.2 if you're lucky. But it's a really, really important concept to understand at a molecular DNA variant level.
Yeah, no, I get that, Rob. I guess what I'm trying to think about here is we need a way of fact checking or like what the behavior really means. Like, are we just collecting noise and we're partitioning noise, or is this really meaningful information? And to get, we could collect, there's a technological thing here that we need to, I'm kind of trying to use genetics to verify this unsupervised learning method. And to really prioritize the behavioral motifs that may be most important for disease. So that's really the goal here. And I will point out that angular velocity, for instance, or temporal symmetry is low heritability. But if you look at the variance of that phenotype, it is, it's more heritable. So
thanks, Vivek. Yeah, that's great. There's a couple of more comments for you in the chat if you want to have a look at.
Oh, yeah, I didn't look at that at all. Okay.
So we've got to move on to the last speaker of our session. I'm delighted to welcome Cora Vacher from Illumina, our sponsor. Cora, are you here?
I am.
Fantastic. Great. Okay, well, thanks very much for this. We'll look forward to your talk, and you can upload your slides. Thanks. Share your slides, rather.
Okay, can you see my slide?
Yeah.
Although everything's gone black for me. There we go, it's black.
Perfect.
So hi, everybody. I'm Cora Vacher. I'm part of the marketing team at Illumina, and I focus mostly or only on projects related to common complex disease, and I have a strong focus on neurological disorder of late to later onset, so anything neurodegenerative and psychiatric, and cardiovascular disorder, and to some extent metabolic. So today I just wanted to give you a quick overview and update on how our solution can really help by doing multi-dimensional approach, maximizing the insight you get from your sample. All of the illustration, or most of the illustration from the talk, are from an ebook that we have published recently. And Renma, if you could put the link for the ebook on the chat, that would be great. So from listening to the talk, I don't think I need to convince anybody on there that omics are the way to go to understand the mechanism of disease. What we are really trying to encourage people to do at Illumina is to talk to us early on in the process, early on in the design, to really include as many omics as possible from the early planning of the project. And by that, we mean, obviously, not all the omics, but the one that we can currently support, which are proteomics, which are the most recent and the one that have been mostly presented during the conference of the meeting so far, genomic, epigenetic, and transcriptomic. And very much this is to accelerate the discovery or novel association, especially to identify association between biological entity, or to find map or prioritize GWAS data. We do see more and more people starting to use the more dynamic omics, so epigenetic, transcriptomic, proteomic, to do identification of relevant biomarker, very often not just one of them at a time, but integrating them, and all of it mostly to toward a goal, which is to try and develop therapy and treat disorder. So one of the points I think that's been quite obvious recently is that everybody does like the newest and most fancy method, and we have seen a lot of energy and a lot of interest going towards single cell and spatial, especially when it comes to transcriptomic and more recently proteomic. Those are really good methods. I mean, I really can't diss it. If you want to have single cell resolution, that's the way to go. But I really don't think this is the end of bulk RNA-seq or bulk protein analysis, because especially when we're looking at human population, or in the scope of this group, the HS group, you will want to look at individual variability. And if you're going to do a lot of sample and a lot of different individual, the solution tend to be much more towards bulk RNA-seq. If you are interested in hearing more from this, we actually have an excellent webinar from Mina Reaton from University College London. She recorded it last year on brain cartography, and she does discuss the advantages of single cell versus bulk, and why the methods are not mutually exclusive, but they really bring a different level of information. And if you do want, the link is at the bottom, but if you do have any question, if you have any link, just drop me an email. My email is on the last slide. I will not add you to any sort of mailing list, and I really do not have time to spam people. So the reason we can really use next generation sequencing in multi-omic studies is that the NGS data really are becoming a universal readout technology. And we do provide solution for a lot of different applications. To do that, we work with a number of partners. Not all of them are on there. I mean, the main one are on them. And that does allow us to provide solution for a very broad range of sample, from what you would expect, bulk tissue and single cell, but also more and more, we do see people interested in fluid, bodily fluid, and extract of bodily fluid. Exosome, I think, at the moment are very exciting, especially when it comes to cell-free RNA or cell-free DNA from methylation. And we do also have solutions that look not only at DNA and RNA, but as I said, more of the structure of DNA around epigenetic. But I mean, most people look at methylation, but there is a lot more excitement recently around chromatin accessibility. And we had a webinar in March solely on epigenetic over 5 days. And again, we have a link. Everything was recorded. If you're interested, please feel free to drop me an email. But where in epigenetic, we do see people starting to look more and more at small RNA for longitudinal study, for things that are much more granular in terms of biomarker, like difference in disease onset, difference in severity, difference in outcome. So we do a lot more of that. Obviously, microbiome. At the moment, I do not know of people using NGS as a readout for metabolome, but I wouldn't be surprised if that comes out in the next few years. And we can do that because we really have a breadth of instrument that covers a lot of different possibilities. So I expect everybody's familiar from either our scanning instrument and our sequencing instrument. And I guess a lot of our audience now really think of Illumina as mostly a sequencing company. And honestly, it's great for discovery. As Denghui Chen presented earlier, if you don't have reference, or if you don't have a microarray already done, you can do low pass. It's really flexible. However, if you're going to have a lot of sample, if you are cost conscious, if you have very time limits on how much you need to, how long you can do your project, arrays are still a very cost-effective way to approach genomic. And we do have arrays for genotyping. We also have arrays for methylation, and we launched fairly recently a new mouse methylation arrays. And it's by bringing all those platforms together, all those approaches together, and statistical data that really multi-omic comes into its own. I just wanted to put an example with a couple of paper, and they are complementary paper from one's from Skene and the other one is from Bryce. And I think they were very interesting. They really show the value of multi-omic, especially when it comes to putting together transcriptomic data after getting loci from GWAS. With Ab on the call, I will not say how many loci there is for schizophrenia at the moment, because I think it's 162, but I'm not entirely sure. But for Parkinson, the International Parkinson Disease Group is now at its 6th metadata, and they do have a massive amount of loci to go through. And both of the paper really show the value of both the approach of bulk tissue RNA sequencing, both in mice and in human. And I think in human, they use mostly data from GTEC. And then looking into single cell to really identify at the cell level which neurons are specifically involved in one disorder versus the other. And they also go down to doing a lot of analysis around different gene sets. And as most of the presentation earlier today and yesterday have shown, there's a lot of pathways that are involved or together or completely independent. And by doing those different approaches and by adding more epigenetic, you can really start deciphering which part goes where. So just quickly, and again, those workflows are on the ebook. So if you're interested, just go download it, it's free. But it's really to show that we now have end-to-end solution for integration of some of the combination. So for example, for genomic and transcriptomic, you do have the choice at the moment of whole genome sequencing and an exome. That is for human mostly, for exome, and adding either whole transcriptome, which is ribodepleted, or focusing more on messenger RNA. For those type of bigger projects, I would think our more high throughput platform like the NovaSeq, like the NextSeq 2000 are more appropriate. But where I really want to put the emphasis is that we are now having really tools for secondary analysis based on our Dragon pipelines, and engines in correlation engine in Bayes, in which you can start putting the data together and extract some integrated information from those data. So if I'm just going to, I'm trying to go quickly because I realize it's soon the break. Really what we're trying to show, and that's what you guys have been showing during this meeting, is that to understand the mechanism of disease, especially after the variant have been identified by GWAS, is that as many layers of omic are necessary, and RNA will help with the fine mapping and the functional analysis and the annotation, whether the QTL or more coding variant. And but they also can provide a lot of information around progression of disease and the different subphenotype of the disorder. The interest of RNA, I think, is that now the tools can really use a very broad range of sample from blood and from tissue or single cell. I would think when you look at methylation, it's even more flexible. You can, methylation it's quite robust. You can use saliva. I'm not sure how you get saliva from a mouse, but in theory you can. You can use cerebrospinal fluid, etc. And what we really see is people are now starting to put together RNA and their DNA and epigenetic at lots of different levels. And one thing I thought was quite striking this year was for people in Europe or accessing European funding, is that for the first time, Horizon Europe, which is the main funding program from the European community for disease of obesity and from disease related to mental health, the call for proposal are specifically asking the project to include genetic and epigenetic. So I really think from a complex disease perspective, we are going to see a lot more of projects, including methylation, attack-chip, and things that are still a little bit more challenging, like ChIP-seq and all the different versions of it, like CanTan-PAG, etc., and the three-dimensional type of analysis. So just as a summary, sorry, my dog is growling and it's really distracting. Just as a summary, when it comes to use cases, we do see more and more multi-omic in diverse disease area. In cancer research, it's starting to come in companion diagnostic, more on the RNA side than on the methylation side. But I guess everybody's heard what Illumina is up to at the moment. And so it's quite clear that methylation is quite interesting in cancer, to say the least, for diagnostic. In terms of common complex disease outside of cancer, integrating gene expression and regulation is going to remain an integral part of understanding the functional genomics and stratifying the cohort into more and more subphenotype or underphenotype for the disorder, and more and more different omics are putting together in lots of different type of fluid to try and identify biomarker or integrated groups of biomarker to really try and predict early on and over the course of the disease how people are going to feel. And I'm bang on 15 minutes. Thank you very much. I'm hoping that I didn't go too fast. Again, if you have any questions, feel free to send me an email. I'll be more than happy to interact.
Thanks very much.